All right, I think we should be all good. I wish there was a way to tell if the attendees are all can hear, but we'll just go for it. All right, good afternoon or good morning, everybody, wherever you are. I'm Oliver Glass. I'm the chair of the Medical Scientific Advisory Board. I want to welcome you to the Scientific Research Roundtable for DYRK1A syndrome. Today, we're joined by Dr. Bernard Khor. Dr. Khor had presented this past July in Texas at our family meetup, so some of you might be familiar with him. And he's going to be presenting some really interesting data around immune dysregulation in DYRK1A syndrome. So with that, I'll pass it off to Dr. Khor. And I'm sorry, I forgot to mention, if you have any questions there, and I know some of us are not as familiar with the WebEx platform, there's a chat function. It's in the bottom right. So you can enter questions in the chat there. And then also, hopefully at the end, we'll have some time, I think, for some Q&A, and then you'll have an opportunity to raise your hand. I think there's a little emoji button that's kind of down on the bottom, and then there's a hand next to it, so you can raise your hand, and then we can unmute you to ask a question. And the way my understanding is that once I unmute you, you have to hit accept in order to ask the question. So just some logistics there. But without further ado, I'll pass it over to Dr. Khor, and thanks again for joining us.
Well, thanks so much, Oliver, and I really appreciate the opportunity. It's really a privilege to talk to you all. It's really a pleasure, and thank you for everyone who's showed up. And I hope you'll find at least some of what I have to say today interesting. I'll give you a little bit of a background. I'm a physician, patient, scientist. So I have I grew up with a genetic disease, hemophilia, so I have a long history in advocating for underserved populations. And I really have been an immunologist. I've been studying the immune system basically all my professional life since I fell in love with it in college. I did my MD PhD at Washington University in St. Louis, Missouri, where I studied early events in the immune system, and then I went to Mass General Hospital in Boston, where I did my postdoctoral work and my clinical training, and that's where I was looking for new pathways that regulate inflammation. I stumbled onto DYRK1A, and that's really been a component of my work ever since. In 2017. I started my lab here in Benaroya Research Institute in Seattle because it really was a unique opportunity to both study DYRK1A both at a cellular and molecular level as well as a translational human level. And the major arm of our lab in studying the human relevance of DYRK1A right now is focused on Down syndrome, but we'd love to expand that to people with DYRK1A syndrome. So with that said, let me share my screen. This worked a little while ago. Let me press that button. And what I'd like to talk to you about is work that over the past decade, I can say that now for real, that we've been doing to really understand how DYRK1A really is an important regulator of the immune response. These are my disclosures, nothing relevant to today's talk, right? And I think the first motivation is really since the data, I'll be frank, that we have in people with DYRK1A syndrome is relatively light, and that's what we're looking to build up. Let's start with some motivation, right? Why is it important to study immune dysregulation in DYRK1A syndrome? Now, the immune system, I think, is really important because, number one, it's an easy way to, the cells of interest are actually easily accessible, so you can get them from peripheral blood, and it's much easier than a tissue biopsy. And we have many tools, right? Because the cells are easily obtained, there's a long history of exactly how we study these cells. And so moving from understanding to therapy is relatively quick. It is not necessarily, that is relatively, that doesn't mean it still takes quite a long time, but it is much easier than in many other organ systems, right? And the other important thing is what we learn in an immune cell. Often we learn principles, right? We learn lessons that are broadly applicable to other cell types, right? So for example, we know that DYRK1A regulates an important transcription, NFAT, and NFAT is important not only in T cells, after which it's named, but also many other cell types. So what we learn can rapidly be adapted to progress our understanding of how DYRK1A might be important in other cell types. And there are many existing therapies, right? Because we know so much about the immune system, there are a lot of therapies, and so therefore there is a large potential for near-term solutions, basically drug repurposing, right? Drugs that have been made for other reasons, and if we understand now that they might be how we might try to understand how they can be used in people with DYRK1A syndrome. This might teach us that sometimes you have a choice of many different drugs, right? Sometimes you have a choice of, say, 10 different drugs, and the way you progress down that is really an order that's determined in people without Down syndrome, right, in the general population. But the order in which it might help people with DYRK1A syndrome might be different. And so understanding exactly what that order is important because it minimizes time that someone's not feeling well, right? And so this might also teach us lessons about prioritizing non-immune therapies. And, you know, I just want to take a little aside, right? Like sometimes we might not be thinking a lot about immune problems in people with DYRK1A syndrome. But I would, you know, I want to kind of harken back to a moment where I was how I really fell down this road. I really kind of stumbled into it just about by accident, right? Which is another example, which is in people with Down syndrome. DYRK1A, as you know, is on chromosome 21, and people with Down syndrome have an extra chromosome. And it wasn't recognized. It really wasn't well recognized that immune problems were really a very prominent feature in people with Down syndrome. And I've had the privilege of being one of the first, you know, one of the early few to really try and proselytize and really advance that, shout that from the mountaintops, right? So that's now very clear that Down syndrome is in part an immune disease, right? And so I think the question, I think it remains an important question of really, are there and what are the immune problems in people with DYRK1A syndrome? Because maybe this is a low-hanging fruit that we can address soon. And there's in terms of what the evidence that we have for that is, you know, there's some evidence. So this is data that Oliver and Anna and Kellen have collected and analyzed, and so this is looking at proteins in the plasma of people with DYRK1A syndrome versus controls. And if we ask what are the proteins that are present in different amounts in people with DYRK1A syndrome, what you find, and let me activate my laser pointer, what you find is there are many immune molecules that appear to be important. Some of the pathways that are the most important, shown here in red, are really interleukin. These are important immune modulators and cytokine signaling, right? So this is basically how the immune cells talk to other cells. And they are amongst some of the most important pathways that are differently altered in people with DYRK1A syndrome, including several cytokines that I'll get to later, whose signaling we think is altered and regulated by DYRK1A. So this suggests, this really lays that principle that there's a really real possibility that the immune system is dysregulated in people with DYRK1A syndrome, and we need more work to really understand what it is, right? Certainly what we all care about the most is really clinical outcomes, but a lot of this is going to be guided by cellular and molecular studies that give us a clue as to what are the clinical questions that we should be asking. So, having said that, what I'll go through today is a lot of that evidence, really supporting what do we know about DYRK1A? What do we know about DYRK1A and immune system, which is actually a fair amount. And what are some of the clinical things we might be thinking about, right? So we know that DYRK1A regulates many important pathways that are important in the immune system. And what I'm going to talk to you about in this first part of the talk is NFAT signaling, which is broadly relevant to many cell types, differentiation of T cells into a pro-inflammatory subset called Th17s. We'll talk about why this is important. These cells are very important in inflammation, they're very important in autoimmunity, and they're very important in the defense of mucosal barriers, which is a fancy way of saying your gut in your lungs, right? And it's also really important in IL-6 signaling, we think. And IL-6 is a really important cytokine. This is a molecule that does many things in the body. And we recently showed that DYRK1A regulates signaling of this IL-6 cytokine through an exciting story that really has expanded our understanding of what we think DYRK1A regulates. And I'll talk about that. Right, so we'll start with, there's work by many people before me that have shown that DYRK1A regulates NFAT signaling. This has been shown to be important in cell types, including bone. And here we show that it's important in T cells, right? So here we're using a fancy flow cytometer that basically asks how much NFAT is in the nucleus. And so the point is when you so basically if it goes into the nucleus, the curve moves to the right. And in the black, you're looking at the T cell that is unstimulated. And when you stimulate it in the red line, now you see that NFAT moves into the nucleus. And when you use harmine, which is a chemical inhibitor of NFAT, you see increased translocation into the nucleus. You're going from 36 to 50%. And NFAT's a transcription factor, so moving to the nucleus means it's going to work, right? And so these are pictures that really tell you the same story. I won't spend too much time on that. But this is the end of a curve, right? So this is a curve showing a time course, and so this is showing in black if you stimulate these T cells, more and more goes into the nucleus, and then that goes away with time. If you inhibit DYRK1A with harmine, this starts earlier, and this is thought to have important consequences in T cell differentiation. So, this data really is consistent with a lot of data in other cell types that shows that DYRK1A regulates NFAT. NFAT is a transcription that is important not only in T cells, but also many other cell types. And so trying to understand how this is an example of how NFAT, learning how NFAT regulates T cells may have many lessons in many other cell types as well. I want to talk to you a little bit about Th17 differentiation, right? So first, I want to explain to you that the immune system really fundamentally has a really complex job. It has to protect you against a near infinite panoply of different pathogens, right? That you have or have not seen before. And at the same time, so it needs to be this really aggressive bouncer, but at the same time, it needs to be tolerant of self, right? And it does this by engaging a very careful balance of pro-inflammatory and anti-inflammatory forces, right? And perturbation of this balance in either direction, right? So too much of an imbalance results in disease, right? So too much inflammation leads to diseases like autoimmunity, and these are really important diseases that we think about. They affect 1 in 6 people, and their economic burden is more than 10 times more than that of cancer, right? Too little inflammation really predisposes people to infection and cancer, and this is a simplified schematic, but these are kind of broad concepts to think, to frame our thinking, right? And this CD4 positive T cell I think about as the quarterback of the immune system, right? It really has an important role in teaching other cells, teaching the whole system, what do we do? Is this friend or is this foe? Are we chill or are we trying to bounce this person out of the joint, right? And it does so by differentiating in either anti-inflammatory subsets, largely represented by Tregs, or pro-inflammatory subsets that include cells like Th17s and others. And more than a decade ago, I set really on this project to ask, could we identify new small molecules that regulate inflammation? And the goal was really to say, can we understand new mechanisms? And because these are small molecules, they're easily made, cheaply made, right? Can these represent new therapies? And so this is a work that I did. I probably started in, I want to say 2011 or so. And what we set up was a really ambitious project that we were told would never work. And we set up a screen using primary T cells. And here we're using murine cells because they're easy to get in bulk. And we stimulated them. We generated ways that we could efficiently generate all these pro and anti-inflammatory T cells of all different lineages in vitro. This is an example showing you that we can generate Tregs that express FOXP3, and then we can look for compounds that increase, for example, the percent of Tregs, shown here going from 38 to 91%, right? And the idea is we generated a platform where we could we successfully queried about 25,000 compounds. What you see here in blue are the negative controls, and most of the compounds have no effect. They cluster close to the negative controls in blue. And then, the dotted line says this is a threshold that we will use to call candidate hits, shown here in pink. And basically, that's exactly what we're seeing. Basically, it says this method works, and there's some technical mumbo jumbo about why this was really cool and unique. I won't dwell on that too much. It was a lot of time, a lot of work. And one of the most interesting compounds that we ended up with was harmine. And one of the things that we learned about harmine and why it was interesting is that it very potently suppresses the differentiation of these pro-inflammatory Th17 cells. So Th17 cells express IL-17, and they're shown here in this pink box in the upper left, right? So this is showing you an example that without any compound, you get about 32% Th17s. When you inhibit DYRK1A, you drop down from 32% to under 2%, right? So potent inhibition of these pro-inflammatory Th17 cells. So is it just harmine? Well, we use a couple of different compounds that look very structurally different compounds that also inhibit DYRK1A. So we use 3 other compounds. They all inhibit Th17. So more inhibition, it goes up, and so a more higher dose of the compound as you go to the right. So dose-dependent inhibition. This is kind of how like a chemical biologist thinks about it, right? So dose response suggests that the response is real. So that's encouraging. And when we use a shRNA knockdown of DYRK1A, so when you use a genetic method to reduce the expression of DYRK1A, we also see less Th17 in blue compared to controls in black. So all this supports, this was published in 2015, and this supports that DYRK1A really helps regulate the generation of these pro-inflammatory Th17 cells. Since then, we've also partnered with Laurent Maher, who's generated a next generation inhibitor of DYRK1A, that's lucitanimine B, LCTB. This is in clinical trials right now. It's got a much better specificity as well as good pharmacokinetics. And you can see also we're efficiently decreasing the amount of Th17s here from 23%. You have to add these two quadrants up down to about 4%. And so you see this dose-dependent decrease, right? More LCTB, less Th17s. If you use an isomer, like what you've done is just made a mirror image of this molecule. And so, it doesn't have a DYRK1A effect, but otherwise the chemical formula looks completely identical. This isomer has absolutely no effect on Th17 differentiation, supporting again that DYRK1A is important for these Th17s. Now, every small molecule can affect other molecules. Yes, we have to accept that. That is true. And so we set out to generate the genetic proof. And so here we generated a mouse where we can specifically delete DYRK1A in the T cells, right? This was a really difficult project, and we just published this this year. And what you can see is if we knock out DYRK1A, shown here in red, you see decreased generation of these Th17 cells. This is done by Matt Maleg and Keegan Mu in my lab. Now, we recognize that there's overlap, and so we wondered if this was because of inefficient deletion. And what Matt showed very nicely was that he could quantitate the amount of DYRK1A that's being expressed. And what he could show is the more DYRK1A there's a linear relationship. The more DYRK1A there is at baseline, the more Th17s that can be generated. And the curves overlap in knockout versus control, might suggesting that this knockout is not 100% efficient, right? But it also suggests a really interesting and new concept, which is DYRK1A is a tunable regulator, right? It seems to be a tunable regulator of inflammation of these Th17 cells. And this suggests that if you express more DYRK1A, you might generate more Th17 cells. And in fact, that's exactly what we observe if we look at mice that have an extra copy of the gene that expresses DYRK1A. They express more DYRK1A and they generate more Th17, supporting this idea that it's a tunable regulator. Okay. Now, we wanted to understand how it was affecting Th17 differentiation, and that led us to understand a lot more about how DYRK1A regulates signaling of IL-6 and other cytokines. Now, the key cytokine that drives Th17 differentiation is called IL-6. Okay, and IL-6 drives Th17 differentiation and many other processes, and we'll get to that later. And the way IL-6 works is that it binds to its receptor, and then this drives phosphorylation of an important transcription factor or protein called STAT3, and it phosphorylates it on the tyrosine residue. So this is an important way of how IL-6 signals, right? And phosphorylation on this tyrosine residue leads to Th17 differentiation. Now, what John Crespino showed, another lab, showed a couple of years ago is that DYRK1A phosphorylates a regulatory site on STAT3. It phosphorylates a serine on STAT3 that increases the amount of phosphorylation on the tyrosine. So it amplifies the IL-6 signal. So this idea, DYRK1A amplifies the IL-6 signaling and therefore drives increased differentiation of Th17 cells, right? So that it raises this question: if we lose DYRK1A, if we knock out DYRK1A, do we lose phosphorylation? Do we get less phosphorylation on the tyrosine? And this is what you're doing here is you're comparing these two columns. If we knock out DYRK1A in red, we get less phosphorylation on the tyrosine, exactly as we expected. And as we expected from John Crispino's data, we also get less phosphorylation on the serine, right? So this is so far a tidy validation of published data, but nothing terribly new, right? We've basically learned that this is also true in T cells. That's interesting. Now, where things started to go really interesting was we know that IL-6 induces phosphorylation not only of STAT3, but also two other important STAT proteins called STAT1 and STAT5. And if we knock out DYRK1A, we see decreased phosphorylation of the tyrosine on STAT3, STAT1, and STAT5. Okay, all of this so far completely expected, right? Now, we knew that in STAT3, we also saw decreased phosphorylation of the of the regulatory serine site. And so the first question was, is there decreased phosphorylation of this regulatory serine in STAT1? And to our surprise, we saw absolutely no decrease in the phosphorylation of the serine at STAT1, telling us DYRK1A does not phosphorylate the regulatory serine of STAT1. And importantly, there's also no regulatory serine on STAT5 at all. And so this really raised this question, is this really all to the story? How exactly, are there other mechanisms by which DYRK1A regulates IL-6 signaling? And this is a long-winded story, so just bear with me because we'll get to why it's so important at the end. There's not a lot of steps between IL-6 binding the receptor and phosphorylation of the STAT proteins, right? So it binds the IL-6 receptor, which is made up of two subunits, the IL-6RA and GP130, right? So these proteins make up the IL-6 receptor. So it binds the receptor, it activates JAK proteins, and these JAK proteins then directly phosphorylate the STATs. And so this raises this idea, does DYRK1A regulate the IL-6 receptor, right? And based on some very old, very loose data suggesting that phosphorylation of the phosphorylation of the IL-6 receptor can lead to its sequestration inside the cell, we asked, does deletion of DYRK1A, loss of DYRK1A, does that reduce the amount of IL-6 receptor? And in fact, that's exactly what we found, right? So this had never been described before. It was very surprising. And what we found is if you knock out DYRK1A, you decrease surface levels of the GP130 subunit in both naive and memory T cells. And this is also true for CD4s, it's true for CD8s, and it seems to be broadly true. Similarly, if you overexpress DYRK1A, you get increased expression of surface levels of GP130, increased levels of the IL-6 receptor on the surface. So again, this idea that DYRK1A is a tunable regulator of the IL-6 receptor, and this is probably what we think is driving, in large part, the tunable response to Th17 differentiation. Okay, and why we think this is now really broken it really wide open, right? Which is Th17s are very important, yes, but IL-6 has even more functions, right? This is a figure from a review showing you what IL-6 does, and you can see here this balance of T cells. Th17s represent this arrow, and the point is there are a whole lot more other arrows. IL-6 does a lot of different things, and so the point is, and I've tried to highlight some themes here, right? It regulates aging, autoimmunity, infection, inflammation, and others, right? And so these are simply the things that my lab works on, and there are many things for many other labs to work on, right? And the point is, if DYRK1A regulates IL-6 signaling, this can have very broad effects on human biology. That's the point. Moreover, what I showed you specifically is DYRK1A not only regulates the IL-6 receptor, it regulates the GP130 subunit of the IL-6 receptor. That distinction is important because GP130 is a shared subunit that is important for many other receptors, for many other cytokines, right? So you can see here IL-27, IL-11, you can read the names for yourself. The point is not these specific cytokines. The point is this explains how DYRK1A can have very profound effects on many different aspects of biology, we think. Right? So what I've shown you in this first part is that DYRK1A is an analog regulator of Th17 differentiation, really leading to questions about how DYRK1A deficiency can affect immune function, right? Can affect risk of infection, can it affect barrier defense in the gut, in the lung, right? Can it lead to lower defense against pathogens, basically, because there are less Th17 cells? And, what does this have to do with autoimmunity? And because it regulates the GP130 subunit, how does this more broadly impact signaling of the IL-6 family cytokines, and how is this important for immune function as well as non-immune function, right? And so in the next, in the next part, I'm going to show you some of the tools and some of the approaches that we've used to try and understand this in humans. My early work, the first work that we've done, has really focused on people with Down syndrome, and mostly because and that's really through our collaborations with physicians and our repository that we set up here at BRI. So one of the great strengths that we have at Benaroya Research Institute is we have long-standing experience in many international clinical trials, international studies, and we're really good at this translational immunology bit. We're really good at collecting samples, preserving them, and studying them. And so I can race through, I can spend as much or as little time in this next bit, but this is really not to focus on Down syndrome, but to show you how the lessons that we've learned might be able to advance our ability to learn about the immune system in people with DYRK1A syndrome. So as you know, people with Down syndrome have an extra copy of chromosome 21. This is the most common chromosomal disorder, affects about 1 in 800 live births, and there is a profound immune dysregulation in people with Down syndrome. So they have increased autoimmunity, susceptibility to infections, and impaired response to vaccines, right? And we know that DYRK1A is expressed in really every cell type that's shown. This is a work from Joaquin Espinosa's group. Every color is a different cell type. And what he's shown that is expressed in every cell. And if you look at people with Down syndrome, in every cell type, they show increased expression of DYRK1A, right? With purple being T cells being amongst the highest expressors of DYRK1A that we've seen. Consistent with this increased amount of DYRK1A driving increased response to STAT6, we see, as we might expect, increased level of phosphorylation of STAT3, right? This is just like we saw in the murine models, increased levels of these Th17 cells in people with Down syndrome. So that's consistent, again, with everything that we expect. And what I'm going to argue is really this idea. One of the things that we've been working on is to leverage this idea of, can we use this profound immune dysregulation in people with Down syndrome as a lens to understand other changes in other conditions. I'm going to show you some examples of how we've done this. But the idea is, can we use Down syndrome as a lens through which we can much more quickly and easily understand which of the changes in people with DYRK1A syndrome are important. And I'll show you how we've used this to make lessons in autoimmunity and immune aging, and how we've really progressed our techniques to look at really cells that are specific for single antigens, which have profound implications for how we study the immune response. And this is really showing you some of our early work where we put together a cohort of people with Down syndrome over a wide range of ages to ask the question, how does the immune system change with age, right? And this is, we're using mass cytometry here, so we're looking at about 70 parameters a cell, about 1 million cells a person. We built some brand new computational and analytic pipelines to analyze this data. And the long story short is this helps us build the highest resolution map of the immune system to date, right? So in people with Down syndrome, and it helps us identify 651 differences in people with Down syndrome, shown here in red, versus without, right? So we can identify many differences. Now we can ask about what does this have to do with autoimmunity, and what we find is every single autoimmune related subset that is up in autoimmunity, which is, say they're red, it's also up in people with Down syndrome, and vice versa. And this includes these Th17 cells that are increased in people with Down syndrome, right? It raises these questions. For example, these atypical B cells that express CD11C, IL-6 is also thought to be important, right? So it raises this question. Are these, right, the prediction would be in people with DYRK1A syndrome, we might expect decreased Th17s. We might also expect decreased CD11C B cells, right? And so those are questions that we might expect to ask. Certainly, we see increased levels of IL-6 in people with Down syndrome, consistent with increased IL-6 signaling. Now, what I'm trying to show here is that this IL-6 signaling is important. Now, in the Down syndrome field, no one had, really 99% of there's very little research in the immune system to begin with, and 99% of that research is focused on interferons, because the interferon receptors are also on chromosome 21. And so here we were trying to ask, are there cytokines other than interferon that are important, right? And so what we had done was we made a map of exactly what markers are dysregulated in T cells from people with Down syndrome. And we asked, could IL-6 also be contributing? And what we found is IL-6 contributes to some, but not all of these features. And in fact, in the same work, we found that interferon didn't contribute to any of these. What we're showing you here is that IL-6 is important, and that also has implications for people with DYRK1A syndrome, right? Because you have altered signaling of IL-6 is what we predict. So this is really already teaching us about potential relevance of DYRK1A syndrome. Does this teach us about how we might expect the naive CD4 positive T cells to be dysregulated in people with DYRK1A syndrome? And what does this have to do with clinically important responses to self-antigens, autoimmunity, to vaccines, to pathogens, so on and so forth, right? I will cut to the chase, which is to say that we've done things that are really difficult, and which is to build maps to show how the immune system changes with age. We've done this and been able to show this idea of advanced immune aging in people with Down syndrome. We can build clocks to show this. We can do this using basically all cell types or only the cell types that are dysregulated in people with Down syndrome. And what this shows is what we can show specifically. This teaches us about other diseases. So when we look at people with type 1 disease, specifically using the immune clock that includes Down syndrome biology as a lens, this shows now advanced immune aging in people with type 1 diabetes. So this is really that concept of Down syndrome as a lens to teach us about other diseases. Can this also help teach us about DYRK1A syndrome? Can people with DYRK1A syndrome teach us about other diseases? Those are the types of questions that we would like to be working on. And I'll just wrap up on some unpublished work showing you how we're pushing the boundaries. I've shown you that we've basically built tools that we can study in great detail the immune system. And we're now pushing this to study cells that are specific for single antigens. And here we're looking at response to COVID vaccination. And we're looking at responses both early and late after vaccination. What we can show is people with Down syndrome show impaired response to vaccination compared to people without Down syndrome, both early after vaccination and late after vaccination. The amount of antibody that they make is lower. And we can show that we can identify the antigen-specific T cells, and we can show that the dysregulation that they exhibit changes with time and is different from the pattern that you see in the non-antigen specific component, right? And we can identify some of the drivers. So again, what we're showing you here is we identified CD38. So this box right here is decreased in the antigen specific component. And what we can show is IL-6 actually contributes to this dysregulation. It drives decreased CD38 in an in vitro model. So I won't go through too much of the other specifics. We're building tools to study the B cell component as well. Suffice it to say that people with Down syndrome exhibit DYRK1A overexpression and immune dysregulation, supporting that DYRK1A, altered DYRK1A expression has important effects on the immune system in humans, right? It suggests that this idea supports this idea that people with DYRK1A syndrome may also have altered immune health, and it's important to understand what that means. And so we're also able to use we also argue that we might be able to use Down syndrome as a lens to quickly understand, especially in smaller cohorts, immune changes in people with DYRK1A syndrome. So, this really kind of wraps it all up. I've shown you that DYRK1A regulates many features of many important immune pathways, and we've shown in our work that it's got human relevance. And we'd like to argue that this supports that it's important to study how immune health is altered in people with DYRK1A syndrome. And so I'm going to wrap up with my acknowledgments. First and foremost, I need to thank participants and their families, which will hopefully include some people on this call in the future, because without them, none of this work is possible. This is showing my family and my lab and cohorts, and we are very community facing. We think that reporting what we do to the community and serving the community is an important part of the mission of our lab. And so this is us at an outreach event for the Buddy Walk for people with Down syndrome. This is my funding. And with that, I thank you for your attention, and I'm happy to take any questions.
Thank you, Bernard. That was really, really interesting. And thank you for walking us through all the work that you've done, not only in Down syndrome, but some of the implications it has for DYRK1A syndrome. I think one of the questions that, may be on a lot of folks' minds relate to, we talk about immune dysregulation in Down syndrome, and you kind of outlined some of the impacts on Th17 differentiation. So would the hypothesis be in its relation to, those with DYRK1A syndrome, that they may have susceptibility to potentially different types of pathogens, kind of given differences that you see in Th17, perhaps, maybe there's differences in viral pathogens versus bacterial pathogens. And have you seen any of that in mouse models where you challenged potentially mice that have Down syndrome or mice that have DYRK1A syndrome with either, like bacterial type infections versus viral type infections, and see how their immune system responds?
Yeah, that's a great question, Oliver. I think, it's a great question, and it's a very complicated question. So the simplest answer would be to say that Th17s, they were originally identified as being really important in fungal infections, right, and parasitic infections. So those you might expect to be very important. But it's also become extraordinarily clear that they have a really important role in gut homeostasis, period. Which is to say that, for example, they have profound effects on the microbiota. So they have, right, and that's really why I think about it not only as potentially important, not only infection, and that's where the experiments you tie out are really important and exactly on the point, and we're looking to do them, but also in overall health, right? Like maintenance. What is the natural tone of the inflammatory state? Because your levels of Th17s, the function of your Th17s has a really important function of that.
And I'll also just mention to any of the participants on the call, if you have a question, you can go ahead and put it in the chat and we can ask it, or you can just raise your hand and we can get you off mute as well.
And you know, I think something that we talked about before, Oliver, that I think is also really important is to be guided by what we see in people, right?
Mm-hmm.
Which is really to say, you know, I think it's just as important to ask families of children with Down syndrome, right? Like, what do you see, right? We think there's something immune related. We have a whole panel, right? We've developed a whole panel of immune related questions, right? What do we see in people with DYRK1A syndrome? Because that could really guide, right? At the end of the day, at my most, to be a little bit cynical, right? I'm not here to cure a mouse of a disease I gave a mouse. I'm in this business to help people, right? And so it's not to say that animal models aren't important. They're very important. But my philosophy has really always been to ground the work in what's human relevant, right? And so let the humans teach us. If the humans teach us these are the top 3 most important things to study, let's focus on that, right? It doesn't mean that you can't do other things, because everything kind of, you know, everything kind of interrelates. Nothing really is a intellectual island in science. But let's focus, right? And so I think an important first question is, you know, what are the immune type? What do we see clinically in people with DYRK1A syndrome? Ask the immune questions, right? Where I think if I kind of go back to my experience with Down syndrome, right? I mean, when people aren't thinking about it, they don't ask the question, right? And simply asking the question can teach a lot.
Yeah, I think in addition to that, you know, oftentimes, you know, when you go to primary care physician, you show up at urgent care ED, and you have a child with DYRK1A syndrome and that has, let's say, a fever or something, could be viral related. You know, oftentimes the physicians will say, oh, well, usually by, you know, the 3rd day, you know, it should start to clear up, or, you know, by the 3rd, 4th day, if they still have a, you know, high temperature, then, you know, bring them back. But, you know, I think from the work that you've done and, you know, some of the hypotheses in the future is that you can't really base, you know, sort of normal regulation of the immune system as it encounters, let's say, strep or, you know, other types of viral pathogens as well. You know, what the time course is going to look like. Because I think, you know, some of the experience from parents and, you know, caregivers is, you know, things can get out of hand and exponentially get worse very rapidly. And that could be due to some of that immune dysregulation. So it would be important to, you know, get some of that clinical data, as you mentioned, and be able to help, you know, support some of the hypotheses.
Yeah, I agree. You know, and you know, it gets at that idea of, you know, precision therapy, right? Like, what is the expected clinical course? And just as important with that, like, you know, how can we improve that clinical course?
Yeah. Anna, you have a question in the chat. Did you want to ask your question?
Yeah, sure.
Yeah.
I was actually writing another one, but I'll start with my first one.
Oh.
I mean, this comes from exactly what you just said, is it's cool to play with mice, but the reason you're doing all of this is to see if it can have an impact on a human population. So my question was, like, are there currently available, clinically available, inflammatory modulators that can cross the blood-brain barrier, i.e. things that can currently address neuroinflammation, if this really is a significant driver in like, for example, kids with DYRK1A?
Yeah, so that's a great question, right? And that is a topic of that's a hot topic, right? As you know, there are many neuroinflammatory diseases. You all probably know it way better than I do, to be honest. But there are a lot of neuroinflammatory diseases. There's a lot of interest in diseases, right, ranging from not only the encephalitides, but also Alzheimer's, right, and many others, and multiple sclerosis, others, right? So this idea of blood-brain barrier that is high on the radar of pharma. if we look at the DYRK1A inhibitors, for example, I think they were designed to be permeable to blood-brain barrier. I think the JAK inhibitors are, but this is the right. But the long story short is certainly the antibody molecules we don't expect to be. Although in the setting of neuroinflammation, we also know that the blood-brain barrier is often compromised, right? And so the normal physics also aren't really on the table. So more things go in. So it's complicated, I guess, but we are well placed to, we're well placed to collaborate with people, and we certainly think to ask those questions.
I think Kate McCarty has a question in the chat. With some of the unpublished data you shared on COVID vaccine response in patients with Down syndrome, is a possible takeaway from that to consider more frequent vaccination?
So this is, although this is a question that's very specific to Down syndrome, I appreciate that. But I'll use it to highlight how we think to bring this back to patients as quickly as possible, right? So certainly we knew about impaired response to vaccination, and so we were part of that group that was really advocating really loud and early for quick access of people with Down syndrome to COVID vaccine, right? Like back when this was all rolling out, if we think back to kind of 4 years ago or so, right? Number two, we're not content with that, and we're trying to understand how to improve the vaccine response. So we're taking certainly the simple approach is to advocate for people with Down syndrome to at least get their annual boosters, right? At least do that, because having the perfect shot matters nothing if you don't get that in an arm, right? Having the perfect drug means nothing if the person doesn't take it. So first, reach out to people. The second is we just got funded for a planning trial. What we're trying to do is launch a clinical trial of high-dose flu vaccine in people with Down syndrome, right? So this idea that the immune system of someone with Down syndrome looks older, we have something, an FDA-approved therapy for people who are older. It's high-dose flu vaccine, standard of care if you're 65 or older. That's 65 in people without Down syndrome years, right? What is it in people with Down syndrome years? Is that 40? Is that 30? What is it? It's safe, it's effective, and by doing it in the setting of a clinical trial, we can simultaneously get the samples to ask, what do we need to be making other than high-dose flu vaccine, right? Because how does high-dose flu vaccine work? We kind of know, sort of, not really, right? But are there other mechanisms? Is there an adjuvant we need to add, a second drug that we need to add to get us to 100% protection? And that's the same kind of approach that we take to approach working with people with DYRK1A syndrome, right? If we see a defect, how is it clinically relevant? Is there something on the table that we can do for a near-term solution, right? Like in the next 5 years? It's not going to be a perfect solution, most likely, right? But then at the same time, set up the studies for what is the more perfect solution that's going to be 15 years down the road. That's kind of our approach. I see Kellen.
Yeah, Kellen's got a question. Kellen, do you want to ask it?
Yeah, I'm just thinking in terms of, because at Combined Brain, we're the consortium for outcome measures and biomarkers in rare disease. Could you, looking at your big heat map for Down syndrome the full picture of the immune system in Down syndrome with age, especially specifically, could you foresee some way to use some sort of immune marker, maybe even just GP130 levels, as simple as that, or even just sort of like the sort of a general, upregulation of these 5 different things as a readout of severity of DYRK1A syndrome?
Yeah, I think that's a really interesting question. I would almost defer to you, Kellen, as really the master in this field, right? And if I understand you right, what you're thinking about is some kind of biomarkers, not only that could either prioritize people for screening for do you have DYRK1A syndrome and or kind of point to severity. I think it's possible. The challenge is this, right? The challenge is trying to understand exactly what those markers would be, because the immune system is very dynamic. Right, and so like if someone has a cold, an infection, then those markers change. They change, right? And so trying to figure out where you are in that spectrum of health is often a piece of that. So I think it's possible, right? Now, the other thing that is also possible is maybe looking at response, right? So most of the studies that we do today are really with this idea. You take a blood sample and you just ask what's going on at steady state, what's going on at rest, right? But you can perturb that, right? And maybe what you're looking for is altered response to IL-6, for example, right? To predict risk. So those actually are quite doable even in the clinical testing, in the setting of clinical testing. They just haven't really been, I think they haven't. It's doable. It's the long story short, right? And so it's something to consider. It's a reasonable hypothesis, I think. And it just needs more data to really say, right, what is the sensitivity, specificity, all that kind of the nuts and bolts of it. But yeah, like if you're right, part of what I'm here to do is also to learn about what are pressing needs in the community, right? And if my expertise can help address those questions, that's great.
All right, I think we have time for maybe one more question. If anybody has a question, feel free to raise your hand or throw it in the chat. And I think, if nobody's going to pop anything up. So what's next for you, Bernard? What in terms of, DYRK1A syndrome research are you interested in? And what are you kind of looking to partner with the foundation for?
Yeah. Well, I think I'm very interested in the role of DYRK1A in immune regulation, right? And I think that's a great fit in terms of my skill set and in terms of the known biology. I think there's a case to be made, and that's, I think, something that's important, right? And something that's therapeutically tractable. And so part of, I think this is what I would say, right? All human research, right? This is the same thing about you can have the best shot and it means nothing if it doesn't go into someone's arm, right? And so the same thing, I can think that is the most important thing, and it might be a really important thing for people with DYRK1A syndrome, but I can't do that work alone. And I say that with all humility, right? I can't tell you what's actually going on in a person until I start studying samples in a person. And fundamentally, that revolves around two things. It revolves around participants, and it revolves around money, right? And that's, if I, to be blunt, that's what it revolves around. And participants, right? Participants, I look at it as something that I have a responsibility to, right? It's an equal responsibility, right? It's to say you are part of families are part of the process, right? Families, it takes work, it takes effort for a family to participate. So it's my job to kind of explain to them why it's important, right? And then it's really kind of relying on, I've learned a lot, right? By simply saying, I need a community to tell me what's important to you, right? Because and what do you, and in that process, I might also learn what are important things to you that I didn't think of that I could help with, right? And I've done a lot of that in the Down syndrome community. We hold webinars like every 3 months or so, and I talk not only about my work, and I talk about my work because I think it's important to say, well, look, if you participated in this work, you should be amongst the first to learn what we learned from it in language that you can understand, right? Because it's important to me, right? Because otherwise, why? More than this kind of aspirational, I help science, which is important, but it's very ethereal. I'd like to give people something tangible. But at the same time also, right, if I can understand that literature, if there's something immune that's related, my lab didn't do it, but I think it has important implications for the community, I'll share, right? I'll help break things down. I'll help answer questions, right? Because to me, it's about being part, it becomes me being part of the community, right?
Yeah, absolutely. Well, I know that we're a couple minutes over. I'll, acknowledge Anna's question as well. She's, asking about sort of what clinical information would be helpful to better understand immune symptoms in DYRK1A
Yeah,
kids.
I'm
I
happy
know
to
you
share
and I
that.
have kind of like talked about that, and I think that's something we're definitely gonna pose to the community to get more information on. We have anecdotal information that the kids get a lot of respiratory infections and don't do well with certain bacterial infections and whatnot. So I think us as a community, we'll definitely, work to gather more data on immune dysregulation and DYRK1A syndrome. But want to say thank you for your time and really appreciate you presenting to our community and continuing to engage in with our community. It was great to have you at the um, family meetup and being a continued presence. So thank you for your time and thank you everybody who joined the call. We really appreciate you listening in and be sure to, I think there might be a survey that kicks out after this to give some feedback on how we can improve the scientific roundtable. So feel free to do that. But I hope you have a good rest of your Friday and have a great weekend, everyone.
Thank you. It's been a privilege. Thank you for joining.